Yes, ma'am.
Yes, also I have started the recording. So I gave you guys one review article. So this review article was published in the Lancet and in 1999. So if you guys are not aware, Lancet is the most cited journal in the world. Okay, and the paper of this journal has a really great impact even at that time, 1999. So that is why, since we are just starting out with our course, we have just finished 2 3 classes. So I wanted you guys to read a paper that is much more basic, not a research article where more complicated experiments will be there, but rather read a simple review article, and that basically explains or describes what is the scope of tissue engineering and what all advancements are possible. So those kind of discussions are being done in this review article. And if you remember, I said in one of my class that Robert Langer, who is currently a professor at MIT in chemical engineering department, so he is the pioneer of this field. So this paper is co-authored by him. So one of the textbooks that we'll be referring to in the course is also written by him and Vacanti. So these are two leading people in this field. So title of this paper is tissue engineering. Basically they have defined what is tissue engineering, and they are saying it is the design and fabrication of living replacement devices for surgical reconstruction and transplantation. So during that time, tissue engineering was only defined with the possibility that we can make a replacement tissue, but now it has become much more that we'll later on understand through various research articles. So let's go through these points one by one. All procedures that restore missing tissue in patients require some type of replacement structure for the area of defect or injury. So this type of therapy accounts for a large part of healthcare resources. These devices have traditionally been totally artificial substitutes like joints, non-living processed tissue like heart valves, or tissue taken from another site from the patient themselves or from other patients, that is transplantation. So it is the problem that they are addressing in point one is still a relevant problem nowadays, right? So people still suffer from those kind of injuries, and that affect very large tissue areas that cannot be healed on its own. It requires surgical intervention. So for those kind of treatment, people either have to go for prosthetics, like knee prosthetics are there. Then people use stainless steel rods and all to fix the bone. Then also there is hip joints. So these kind of artificial prosthetics, or you can say methods are there, substitutes are there to replace the injured area. Or people have also started using the pacemaker or the heart valves. And the last thing that is there is transplantation, the whole organ transplantation or tissue transplantation. But uh, it you know very well that it is very very difficult for people to get a donor who is willing to give his or her organ. So it's a very difficult thing in place. So in order to circumvent the limitations, Mumbai, can you mute yourself? Some voice is coming from your end. So yeah, I could mute him. So as I was saying, in order to circumvent this relevant issue, and it's a very very relevant even at all times. People need very good healthcare alternatives, right? People need to live longer. So that is why tissue engineering has come up, where the field promises to make artificial tissues, and that can be made available to clinicians so that they can use it to replace the lost tissue and promote healing of that particular area. So these tissues are constructed to meet the needs of each individual patient. So now through additive manufacturing techniques, um, we can actually make grafts or substitutes which are only relevant to the patient injured area. Say a bone has cracked in such a way, a very small injury is there, right? And in order to fill that particular gap in the bone, you need a particular size or a particular shape of a stainless steel implant or titanium implant. So those can be made nowadays. There then it was just a concept that it was being introduced. Now it's a possibility, we can do it. So what is tissue engineering? Tissue engineering basically is an interdisciplinary field. You must remember. So if any of you guys want to pursue research in this area, you should know that not only your knowledge in biology is going to help, you also have to learn about material science, then chemical engineering like mass transfer, heat transfer, then you'll have to also learn more about cell biology, and then you have to interact with the surgeons in order to understand what kind of uh, tissue they really want, how surgeries can be made much more easy for the patients. So these kind of inputs from clinicians are also important in order to make a successful artificial tissue graft that can be used directly on the patient. So it's an interdisciplinary field. Only the knowledge of one area is not sufficient. You have to work in the interface of various areas. So it is an interdisciplinary field which applies the principles of engineering and life sciences. So in engineering, mechanical also come, chemical engineering. If you are working in nerve tissue area, then bit of electrical engineering, because there are various kind of polymers that are conductive in nature. So those kind of flow, if you want to study how the electrical influxes will flow through artificial tissue. So engineering in every aspect you can think of can be useful. And life sciences, as I said. Now these two principles you have to use in order to make biological substitutes that restore, maintain, or improve tissue function. So I think in our first class we talked about a definition similar to this. So now you can understand from where that definition came from. So this is the original definition of tissue engineering. So some prominent examples of tissue engineering were already there, like I have written. In 1975, there is a scientist called Chick and a few of his colleagues. They were able to encapsulate pancreatic islet cells, that is beta islet cells, in semi-permeable membranes. So we have already talked about it, right? Encapsulation of beta islet cells. So this helped in controlling the glucose level in patients with diabetes. So this proof of concept was already given in 1975, and now if you search internet, I'm sure there is some commercial available treatment available being used in clinic nowadays, I'm sure, but it is not yet affordable for every economically section of the society. Our job now is to make such kind of tissue engineered therapies that are also affordable, so that even like people belonging from a lower middle class or even the below poverty line person, he or she can avail that treatment. So that should be our target. And also replacement of skin has been done by using cells, and they are encapsulated in collagen or collagen glucosaminoglycans, that is GAGs. So these kind of within this composite, within this natural polymer composites, the cells were encapsulated, and that are showed as a suitable skin substitutes. So these skin substitutes they have been shown that they can promote regeneration of the injured area. So these were shown in 1980s. So these are some proof of concept studies that have demonstrated that it is possible. These type of therapies are possible. Now, a recent survey of tissue engineering, I'm talking about during 1990s. Okay, so this is not a data of now. I've already shown you the data of now, I think, how the tissue engineering market is growing, but this is a data given from 1990s. So according to the paper, during 1990s, tissue engineering was an emerging industry. Okay, and the companies which are there in this industry, the total capital value exceeded 3.5 billion US dollars. Okay, so that's like a huge amount during those times, and they projected that this industry will grow at least by 20%, like every year, and the total expenditure that this industry wants is around 450 million dollars US dollars. So much money has to be spent, so many people, like over 2500 scientists now almost in every IIT or ISC or ISSS. So there you will find at least one faculty who is working on tissue engineering. So now even in India and abroad, everywhere you will find one or other faculty is working on tissue engineering or a similar area to that. So that's why more and more scientists and more students are getting involved in this area. So it has been estimated that the total market potential of tissue engineering products in the US alone is 80 billion US dollars. So now the numbers will obviously be much more. But think about during that time, this field has this much potential. So now it has much more. The primary goal of all approaches to tissue engineering is the restoration of the function. All right, so restoration of the organ function or tissue function through delivering of living elements, and which becomes integrated into the patient. So your objective is to restore the function of the damaged tissue by providing some living elements like cells or some growth factors, cell plus growth factors, or only a polymer scaffold or combination of the cells. Like you can make combinations as well where cells, polymer scaffold and growth factors are there. So these kind of living elements, and now people are also including other things like DNA molecules, RNA molecules or nanoparticles. So a combination of them and their function, their combination of these should play an important function, that is to restore the function of the damaged tissue. That is the primary goal. Some techniques of tissue engineering, they rely on matrices alone. So here matrices, I mean scaffold, artificial scaffold, which can be made of a polymer or metal or ceramic. So we will talk about that in more detail, but here just to give you a brief idea, the matrices that I'm talking about is the scaffold, which can be made up of different non-living materials, or they can rely only on cells. So as you have seen, while people are delivering encapsulated beta islet cells, so here the cells are only doing the therapeutic effect. The surrounding encapsulation material, it has only it doesn't play a very significant biological role, but to protect the cell, only that much it is doing. It is not directing any cellular function, etc. But in matrices, people design a matrix in such a way that it promote either cell adhesion of the surrounding neighboring tissue, okay, or they release some kind of drug. So this biological function is there in these kind of matrices. And as I already said, a combination of these two, or and now like nanoparticles, etc., you can combine and make a tissue engineered product whose job is to restore the function of damaged tissue or damaged organ. And in tissue engineering devices or in tissue engineering medical products, stems that we know already, there you can use controlled drug delivery methods. Okay, targeted drug delivery methods you can use to deliver growth factors. Okay, so why you need to use growth factors and all, we'll talk about that in our discussing in our upcoming discussion sessions. But in simple, growth factors or cytokines, they are cell signaling molecules. All right, so when you create a gradient of these growth factors, the cells can sense it, and they will try to migrate towards the source of that growth factor from where it is releasing. So in that way, whatever signal it is getting, it will do some kind of a function. So when you want to trigger a particular type of function, you will use a particular type of growth factor. Okay, so this kind of engineering you need to do in order to get a overall therapeutic effect. So growth factors like VEGF, vascular endothelial growth factors. So these VEGF growth factors, they have been used to promote angiogenesis, that is growth of new blood vessels from the pre-existing one. So angiogenesis is a very important step, you can say, that happens uh, towards the late stage of wound healing process, even after, even in a cancer or tumor formation, how the growing cancer or the tumor is getting blood supply. It is getting blood supply through angiogenesis. Okay, so the cancerous cells, they release the VEGF growth factors and other growth factors that promote angiogenesis, because of which new blood vessels grow and completely supply the tumor with all the nutrients and oxygen. So their overall effect is negative for the person, but you can use this process to create a overall positive effect, positive therapeutic effect as well. Or you can also use these growth factors to aid in completely new tissue regeneration. Okay, it is tissue generation. So you will study in the next class only, that every tissue has a regenerative potential. Every organ, I can say, uh, they can proliferate, but the rate of proliferation, the rate of cell production is different. It varies from species to species, it varies from tissue to tissue. So you need to consider these facts while engineering this kind of tissue engineered medical products. So while you're making this kind of product, you need to focus on three main sub areas. One is, you need to decide what kind of matrix you want to use. Okay, uh, and then you need to decide what kind of cells you will use, and then if you're using cells, how will you culture the cells so that you get around million to billion numbers, and that number is called a clinically meaningful cell number. So you need to grow that many cells. So how will you do that? So you need to consider these three factors. Now if we look into the matrices or the scaffolds, let me use another term that is very frequently used in tissue engineering, that is scaffolds. So these scaffolds, they are used as substrates. Okay, within which substrates for cell adhesion. Okay, so there are two types of cells in our body. One type of cell can adhere, other cannot adhere. So can you give an example of non-adhering cells? Cells that do not need to adhere on any substrate, and yet it can still do its function.
Egg cells.
Egg cells. So you mean the female gamete, right?
Yes, ma'am.
So they actually,
Blood.
yeah, Shambhavi, you said blood cells, right? Your
Yes,
voice was,
ma'am.
so you are correct. it is the blood cells, the RBC, WBCs. They are always floating in our blood. They don't adhere anywhere. Even all the other cells, cell types like platelets and all, so they are also non-adherent. And all other cells except blood cells are adherent types. They need a substrate, even the egg cells. So after fertilization, it needs to go and graft on the surface of the uterus, right? Endothelial lining is there on which it will graft. So it needs to adhere. Only the blood cells is there which doesn't adhere. That is why when you are doing cell culture for non-adhering cells, the media requirements, etc., becomes completely different. The steps that you will follow becomes completely different. Whereas for adherent cells, you need flask where you need a substrate. So, do you guys know what kind of coating is there in the cell culture flask on which you grow cells? Like, is any of you are familiar with mammalian cell culture techniques? No? All right. Maybe you can look it up. Yeah, you were saying something. Shambhavi, you were
No,
saying.
ma'am. I was saying no.
No, no. All right. So I was saying that maybe you guys can look it up. What kind of coating is there in cell culture flask? And it's a very very basic question. Um, if any of you guys will be doing any project or anything like that, you should know uh, of how does cells like fibroblasts, etc., are cultured, and what kind of coating is there on the flags that are used to culture them. Right, otherwise we'll discuss later the tissue culture techniques, but you guys can look it up. It's a very interesting task for you. So as I was saying that scaffolds, they are either used as substrates or they are used as material for encapsulating mammalian cells like the beta alert cells. And these scaffolds, they can be made up of either synthetic material or natural materials. So now in the area of material science, or the engineers who are working in the material science domain, they have made materials that are called semi synthetic, which kind of those materials, some semi synthetic materials, they kind of combine the properties, kind of, okay, not completely, they kind of combine the both the positive properties of synthetic materials and natural materials. So some few synthetic materials here, only the polymer names are there, but the synthetic materials, metals, ceramics, or composites of these are also possible. So here example, PLGA or polylactic glycolic acid, or PAN, polyacrylonitrile, or PVC, polyvinyl chloride. So these are few examples. When we'll talk about biomaterials, we will see, we will actually go into details about polymers and various examples of their etc. Whereas in natural materials, they are basically those materials that are derived from a plant or animal source, like collagen or hydroxyapatite. Okay, it's an inorganic calcium phosphate crystals, but since it is present in our bones, our bone contains major amount of hydroxyapatite, so that is why it is given as natural materials, but it is actually a ceramic. Okay, we'll see that again in the biomaterials discussion. Also alginate, although it is not taken from animal cells, but they are taken from an algae. So these are few examples of natural materials. The advantage of synthetic material is that we can actually control the material's strength. By strength, I mean the Young's modulus, right? The elastic properties, etc. Then they are very fairly easy to process. It means processability is easier. We can control their degradation rate. the microstructure. Microstructure, I mean the porosity and permeability. Okay, so these things we can control. You will understand gradually why we need to control all these 4-5 things that I mentioned, but I think you can understand why strength is important. Other degradation microstructure, we will come to it later. So if the substrate is not having some strength, the cells, they will not adhere. So bone cells, they need a surface with very high Young's modulus in order to show its functionality. Whereas if you look at the cells from the liver, that is soft tissue, there the substrate that needed is comparatively low Young's modulus substrate will also work for its functionality. So this kind of thing, if you want to control cell from cell type, then we need to control that what is the strength of the material that we are using. So you need to do some characterization, and for doing that you need to know a bit about mechanical engineering as well. So as I said, it's an interdisciplinary field. Now you might be able to connect. Okay, whereas the advantage of natural material is that these materials are actually those materials that are naturally available in the ECM. Okay, so the cells recognize these materials because they were interacting with them in vivo. So collagen is present in vivo in the ECM. So if we use extract collagen from any animal tissue, or actually animal tissue you can do, like rat tail, etc. So if you extract collagen from animal tissue and encapsulate cells in collagen, or grow cells on the collagen substrate, on the surface of the collagen substrate, the cells will easily recognize it because the cells already have integrin receptors for the domains, for the cell binding domains that are present in collagen. So it is easy for them to recognize the substrate and adhere, and then subsequently uh, you know uh, step by step show cellular fate processes. That is why natural materials are also intoxic, like they are not toxic to the cellular growth. And these materials have naturally the adhesiveness. As I said, the cells already have the integrin receptors needed to recognize the cell binding domain that is present, say, in collagen, and thus easily adhere to it. So this kind of property is naturally not there in synthetic materials. So people have to add those properties through small peptides like RGD peptides. They have to put it as a side chain molecule to the entire backbone of a synthetic polymer. So these kind of material engineering things people have to do in order for that material get be compatible with the cell. The term biocompatible is a very important term, okay, that I just introduced to you guys. So in order to make that material biocompatible, they have to engineer in such a way that the cells can recognize the surface and show the cellular fate processes. We will formally describe the definition of this term in a later discussion. So one research area involves synthesis of tissue regeneration templates that integrate the properties of both natural and synthetic materials. So if you recall, I said about 7 synthetic materials. So these materials kind of integrate properties of natural and synthetic materials. All right. So this is a new class of material. So these materials haven't yet got FDA approval. A lot of research is going on where people are using this material in order to make artificial tissue grafts or drug delivery vehicles, etc., in order to show how biocompatible or how non-toxic they are. Means they are trying to show its properties, beneficial properties. So in future, soon these material will have FDA approval. Otherwise, collagen, gelatin, alginate, these all are FDA approved materials. So in order to like while you are choosing a material, okay, for making substrate and for making a scaffold, you need to make sure that that material is biocompatible. So biocompatible means it should not be toxic to cells, all right. It should allow the cells to do its intended function, or overall it allows your product to do the intended job that you made. So the material that you have used for making a catheter, all right, it should be such that it should not promote microbial attachment, like it should not promote the formation of biofilms. If it does, then the material is, or that catheter is not biocompatible. So based on your application, whether or not your product means overall product is meeting the requirements of that application. So that is how we define the term biocompatible, right? If it's not clear, you guys can let me know. I'll explain later. Explain again. Okay. Another thing that one needs to take in mind, that the matrix or the scaffold that you're making, it should be able to meet the nutritional and biological needs of the cell population. All right. So as I said, that we need to make sure, or we need to control the porosity or the microstructure of the material. So if the matrix or the matrix is not porous enough, it will not allow proper diffusion of oxygen or nutrients to the cells. So the cells, whatever you're encapsulating or you're seeding on the surface, at the center of your 3D structure, the cells will start undergoing, hypoxic condition, that is oxygen deficiency, or will experience malnutrition, or you can say nutrient is not getting enough to those cells. Then automatically the cells will switch on the apoptotic mechanism and start programmed cell death. So these kind of things will start happening. So you need to make sure your matrix is such that it is able to flow the oxygen and the nutrients to each and every cell in your product, tissue engineered medical product. Then it should be biodegradable. So now the trend is, in tissue engineering field, the trend is whatever product that you are putting inside or on the surface of the body, if it's biodegradable, then you do not have to worry about doing doing another surgery to take it out. So usually when people put titanium rods or stainless steel rods on a fractured bone, right? So what happens? People have, after a year or 6 months of recovery, doctors schedule another surgery to take it out. But for tissue and medical products, if we use a material that is biodegradable in nature, and it makes non-toxic byproducts like carbon dioxide or water that can be easily, exhaled out or flows out through your urine. So if these kind of products are being made, then you don't have to worry about it taking out. So you need the product only for the time of healing. All right. So if the wound takes 6 months of healing, so you need that product to stay there to provide therapeutic effects for 6 months. And once the surrounding tissue, the cells in the surrounding tissue are able to, regenerate the area completely with the correct mechanical strength, the surrounding scaffold is not at all required. The cells will only release the enzymes, proteolytic enzymes, and cause biodegradation of the polymer or your metal, etc. and that so the byproducts will definitely flush out from your system, either through your kidney or through your lungs, etc. So this is the trend. That is why people want their mattresses to be biodegradable. It is much less painful for the patient who is undergoing this kind of a treatment. Then it should also have a good mechanical characteristics. I mean, the Young's modulus, etc. And it should be the mattress that they're making should be adaptable in creating highly specific shapes specific to the injured area. So if you need a small tissue, small mattress to fit a small size defect, then you should be able to make it. If you need a very large volume of if you need to fill a gap of a very large volume, then you need a large shape. Also, the shape can be irregular, right? According to the defect, you need to engineer it. So this kind of material, if you're using this kind of material that is giving you the flexibility of making any kind of shape, then it is also beneficial for the healing purpose. So in this figure, you see the names of all kind of organs are there in our body, and these names are basically highlighting for how many tissues people have attempted to make tissue engineered medical products, and have shown as a proof of concept that proper healing has taken place. And you can actually divide the field of tissue engineering into the organ type, like there is skin tissue engineering, liver tissue engineering, lung tissue engineering, heart tissue engineering, brain tissue engineering, actually nervous tissue engineering. So these kind of names are also there, but the target is to make a product that is able to promote healing to that particular organ if it is gone some kind of an injury. So this is the figure. Now for some applications, the mattress alone is sufficient. All right, say for bones, if you make a mattress that is having a very good mechanical property matching that of the natural bone, if you have it, and it is biodegradable, then and also the material is such that on which the cells can easily adhere, if these things are if these factors are met, then it is sufficient for healing of the bone. But for some kind of tissue defect, you need the cells as well. All right, like the beta islet cells. Without beta islet cells, through mattress alone you cannot deliver the insulin hormone. Yes, you can do that, just load insulin hormone into a capsule or anything like that, but it is a temporary thing. If you encapsulate the cells will grow it for a very long time, right, and steadily produce insulin whenever the body needs it, because the cells have their own sensing mechanism, right? They can sense how much glucose level is there, and whenever needed, insulin is produced, just like what happens normally in our pancreas. So we are trying to mimic that. So always remember, in tissue engineering we have to first understand normal anatomy of the organ or the tissue, and then people try to mimic that. All right, because if you don't mimic, if you don't understand the natural healing process or the natural process that is going on in the body, you cannot make a product that is sufficient, or that is sufficient to regenerate, or for replacing an organ function. So that is why, for some applications, cells are very, very important. The cells, whatever cells that we are using, they can be either sourced from patient themselves, that we have already seen in autologous chondrocyte transplantation, or it can be from a relative or some other individual who is willing to donate his cells. So these are allogenic cells. So in some cases, cells from animals are also taken, that is xenogenic cells. So based on what is the cell source, you can categorize them. So another potential cell source is the embryonic stem cells. So the use of embryonic stem cells hasn't become legal actually, means it hasn't, all the studies that you'll see, it is all in the lab scale. It hasn't gone up to clinical stage yet, because a lot of ethical issues also comes into role. People will start asking like whether it is ethically correct or not, morally correct or not, using this kind of stem cells. So but once people sort it out, scientific point, use of embryonic stem cells is a way to go, because these stem cells are, only have the potential to differentiate into multiple type of cell, but you have to know how to control that differentiation. So that is a challenge, as I've mentioned in point 1, that you need to learn how to control the differentiation of the stem cell population into particular cell type. Usually what happens when people use stem cells, even a particular type of growth factor or cell culture media is given, still you will see that multiple types of cells are there in your flask. So you need to be very precise in using your concentrations of different things, in order to control that only one type of cell population is there, so that you get a pure culture, not a hybrid kind of a thing. So another challenge is also that to make a pure stem cell preparation, because what happens when you isolate stem cells from a particular tissue, be it from fetus or from adult, along with stem cells, the other cells surrounding it, like fibroblasts, etc., they also come out. And through naked eyes, you can't differentiate. So through proper characterization at the molecular level or at the cell surface level, you have to recognize those cells, and then you have to figure out how to separate other cell populations from stem cells. This is a challenge. And also you need to also methods need to be devised to reduce cell adhesion during culture. All right, so if you can make sure that most of your cells, means it depends on the cell type. I'm not saying that it is, you don't adhere your cells, but if the cells need to be adhered, you need to. But reduce in the sense, sometimes the integrin receptors on the cell surface have the ECM proteins attached to them. So integrin receptors are limited. So few population of integrin receptors, they are occupied. The rest are empty. So when you're using those kind of cells with partially occupied integrin receptors, then you won't be getting maximum uh, efficiency from the cells to adhere on your substrate that you have made. So this kind of issues can come, and if your cells are not adhering properly, then the cellular photosynthesis will not begin. All right, then also processes are needs to be optimized, that helps you to produce these primary cells in a very large number, because primary cells take a long time to culture, and even to culture 1 million cells, it may take 2-3 weeks, and sometimes you may not have that much of time. So processes, bioreactors, etc., need to be engineered, all right, in order to help these kind of processes. So another potential cell source is the progenitor cells. So what are progenitor cells? We'll talk about them when we will study about particularly about the stem cells. But for now, you think that these are unipotent cells, means they can only differentiate into one type of cell. So embryonic stem cells are multipotent. So if you give correct environmental conditions, they can differentiate into multiple cell types. But progenitor cells, they are unipotent. How progenitor cells are made, etc., we will discuss in detail when we talk about stem cells. So these are another potential cell source. So, and these progenitor cells are actually present in almost all organs, in those actually in those organs which can regenerate themselves, like liver. Although liver takes very long time to regenerate, but it can regenerate. And regeneration, or is all only possible if there is a small population of stem cells present. Skin also have stem cells. So people have actually recognized small oval cells that are actually the progenitor cells of the liver, which can turn into either mature liver cells when required or into bile ducts in culture. So in vivo, these stem cells usually stay dormant, and they get activated only when it is needed, otherwise they don't. So you will understand why again when we discuss about stem cells. And lastly, what we can do is to create universal donor cells. So you don't need to worry about taking cells from the patient, like or taking cells from its relatives, etc. Also, you don't need to worry about the stem cells because these cells have a lot of ethical issues connected with them. So why not create a universal stem cell line? Where when so these cells, whenever it can be put into anyone, and these cells won't be recognized as foreign in the body, they won't display any immune response, they won't be rejected by the body. So if you can engineer this kind of universal donor cells line, then also you can use them as a potential source for your tissue engineered medical products. And one way of doing it is by masking their MHC complexes, right? Major histocompatibility complexes that are present on the cell surface. That helps the uh, surrounding macrophages, etc., recognize these cells as self or non-self. So if you can mask it, then they won't recognize as non-self, and the cells would be left alone to do its own function. So these are this is a discussion on cells. And another sub area that we need to focus is optimizing our in vitro culture systems. So you need cells. So to get cells, you need to culture them first. So how would you culture it? So in lab scale, it is easy. You need a limited amount of media, you need limited amount of different reagents, but the cell number that you will get is too less, and that is not sufficient if you are making um, large scale in large scale, like in a factory kind of setting. So for that kind of a setting, people are using or engineering different kind of bioreactors. All right. So through bioreactors, you can create large volumes of cells, and these cell volumes are much greater than what you will get from your lab scale cell culture techniques. But it more or less um, things principle of cell culturing remains same, only uh, scaling up has happened, the media, etc., is required more. And in lab scale, we don't worry about mass transfer or heat transfer of things like, we know a small volume of media, it will maintain 37 degrees Celsius if you keep it in an environment that is at 37 degrees Celsius. But in large reactors, you need a continuous heat coils, etc., that continuously flows around warm water to keep the entire say tube or entire container at 37 degrees Celsius. This kind of heat transfer engineering people have to do. Or mass transfer means you need to make sure that uniform distribution of nutrients and oxygen is there in a very large volume of media. So so people use propellers, etc. And also you need to make sure that it is not harsh. It is not harsh for the cells, means they should not experience too much stress, shear stress, because of which cell lysis can happen. So all these things people have to consider while engineering this kind of bioreactors that gives a very large volume of cells. Then only you can create at a time, say, hundreds of these products, and then you can help hundred of these patients. But in lab scale, you can only create a product for only one person, so that is not very economical, right? um, so one example of how use of bioreactors have actually helped in making a tissue engineered product is given. So cartilage, cartilage is a tissue. It is a connective tissue, in fact. And for making artificial cartilage constructs, what people have done, people have cultured the chondrocytes on a 3D polymer scaffold. Okay, and the culturing took place in a bioreactor with and they made sure that the nutrients and the oxygen were mixed well, and the motion of the fluid such that the mass transfer of the nutrients, oxygen, as well as the flow out of the waste from the cells, it is happening uniformly. All right, they made sure through this bioreactor. So because of this, they observed that the cells were stimulated in such a way that it produced 60% more collagen in the matrix. All right, the cells made 60% more collagen than what you would expect if you do it in a lab scale setting, or when you do it not in this kind of a culturing system. And also the gags, these are again the ECM protein. It is the content of gags increased by 125%, and the overall cartilage construct, it became much more thicker, around 30% more thicker, because of which the construct has a better mechanical property. All right, so the mechanical property, as I said, it should match the mechanical property of the surrounding tissue. So you are going to implant it on the injured area. So the surrounding tissue and the implanted tissue, they should have a similar mechanical property. Otherwise, engraft won't be happening very well. All right, the cells that you have given, they won't grow very well. So these kind of things improved mechanical property, then secretion of the ECM proteins by the cells. These all improved when people used a bioreactor kind of a system to make tissue engineered medical products. So all this sounds very interesting, right? So this was all about this paper actually, where they have discussed the uh, scope of the field. They have discussed what all three important areas that people need to focus on according to them. Now the trend is now that actually the research problem is uh, on like simple proof of concept is there, but you need to actually go to the clinical scale, right? That you can make a large number of products so that you can affect large number of patients and make this therapy much more economical. And not only that, uh, now like in 1990s people were happy with only cells and the matrices. Now people want to incorporate the network of blood vessels, that is vascularization, then the network of nerve supply, that is called innervation, or network of lymphatic system. So these kind of things also people want to integrate. So these are the future areas of tissue engineering that people are working on now. All right, and I hope in next 10-20 years we will have much more advancement in this area. So any questions till now? Any questions?
Hello, ma'am.
Yeah.
What is the main technical problem we are facing when we are constructing whatever you say, ma'am, whatever the research fields are said on trends, technical problem?
So you're asking a very, very general thing. So let me think about it. So if you're using a known cell, right, like a primary adult cell, then cell culturing is not a problem. Even the stem cell culture is not a problem. If you are using a material that is very well known, whose synthesis is very well known, uh, and you know all the characterization, how to measure the mechanical properties, etc., then also the making of that material or modulating the shape of the material is not that a difficult job. difficult job is to combining these two. All right, combining these two means the cell that you are choosing, it should be able to grow on your material of choice. So you have to think like that. All right, and when you are going to implant it in the body of a patient, then you have to think whether it's going to be toxic or not. So these are the small, very small challenges that we are facing. Then later more challenges are there, like whether after implanting the tissue engineered medical product, it has become supplied with the body's blood supply or not. Because if it's not, after a few weeks your implanted graft will slug off, it will simply peel off. It should actually very well uh, very well get connected, like become one with the patient's body. And if you are using, say, artificial skin tissue, then people also want that it should become aesthetically pleasing, so that it shouldn't leave behind any scars. So it varies from tissue to tissue type, organ to organ type. The challenge becomes at the interface when you are trying to combine two and three, like various elements together, and trying to see whether overall they are giving your desired effect. All right, there the challenge comes. Any follow-up question, or you satisfied with this answer? Okay, all right. So any other queries? All right. So I I think so. If you guys haven't read this paper, I ask you guys to have a read. It's a 3-page paper only, and whenever you get a free time, please have a read. So during our quizzes, I had a query from one of you actually about the syllabus of our quizzes. So like we will have our next week, we will have our quiz in next week Friday. All right, I have already emailed these things to you. So for the next week, whatever syllabus that we have covered, say from today. So last Saturday Sunday we had our quiz, right? So on Saturday we discussed few topics. So after quiz 1 is completed, all right, and before quiz 2 is going to start, so in between whatever syllabus has been covered, that will be the syllabus for your quiz 2. Got it? So in this way your quizzes will be there. You don't have to worry about reading the entire syllabus for a particular quiz, but I would recommend that you don't forget at least what you have studied for quiz 1, because everything in this course is interrelated. So if sometime you need to go back and revise your concepts in the middle of this course, please do it, because if you don't, then you will not be able to catch up the advanced concepts that will be coming up. Then the things will become difficult for you. Now it's introduction is going on, so things are easier, but now few more topics are going to be a bit more challenging, and so then you have to be bit more thorough with revisions if needed. If you are able to grasp it in one go and remember it, then no need of revision or anything. So that is how your quizzes will be there, and that is the syllabus of your quizzes. So and also have a read of the papers that we'll be discussing, because at least one question will be there, all right, in the quizzes. And I did every one of you get the marks of quiz 1? You have received it all, or did is there anyone who didn't receive the marks? All right, so are you guys satisfied with your
We
marks?
received a mark.
All right, are you guys satisfied with the marking, or is there any query on the solution that I've shared? Any question of the quiz that was not clear to you? you want me to discuss? Is there anything like that? No, right? All right. So thank you all for joining. I'll see you guys on Friday. So on Friday, or Saturday, I'll see. Maybe on Saturday we will have a quiz too, because not much syllabus has been covered. So on Friday we will have our class, normal class. On Saturday we will have a quiz too. All right. So then I think enough syllabus will be covered, enough content will be covered for a quiz. All right. Thank you. If you have any questions, you guys know.
Ma'am?
And yeah. Yes, Janvi.
Ma'am, on Saturday there is 22 July. Uh, my task performance paper on that day. I already told you.
Okay, you have a paper, no? All right. So maybe we can.
Ma'am, 21 is fine. Like
21
Friday
is
is
fine.
fine.
Friday is fine. Okay, fine. We will have it on Friday. Let's see. All right. I'll inform you whatever is the case. But you don't have to, wait for my reply. But the mail or anything, you just continue with the reading. Whatever content is being covered after the class, make a habit to have a look at the notes. You guys know I shared the notes with you guys, so you guys can have a read, so that you don't have to worry about studying at the last moment. All right. So thank you guys. I'll let you guys know about the date of the quiz too, over email as well as in WhatsApp. So you guys can now leave the meeting. I'm stopping the recording.